Hey class, welcome to the voiceover for our next lab. In this lab, we're going to cover 3 different labs from the lab manual, labs 5.4, 5.5, and 5.6. So we're kind of carrying on with our theme of doing differential tests. Remember, the point of differential tests is to characterize bacteria and identify specific features about these bacteria that help us eventually identify what that actual bacteria is. And so the focus of this week's test, these 3 different experiments, is to look specifically at bacteria that have the ability to respire. So I included the questions that I think are kind of valuable for you in these 3 tests. They're right here. All right, so let's remind ourselves a little bit about what the point of respiration is and really just kind of energy processing in general. Remember that bacteria are, many bacteria, especially the bacteria anyway that we look at and are of medical importance, are heterotrophs. They have to get their energy from some reduced form of carbon, and typically that's some type of carbohydrate. So of course the carb of choice and the carb that we look at the most and most bacteria can use is glucose. And so bacteria's job, and our job too, really, I mean, we're kind of the same as bacteria in this regard. We need to take the chemical energy that's stored in the bonds of glucose, and we need to convert that chemical energy that's in glucose to potential energy in ATP. Bacteria do it, we do it, all living things that are heterotrophs anyway do this. And there's 3 different ways that bacteria can do this. We talked about fermentation last week. If you recall fermentation, the kind of defining feature that determines whether or not it's fermentation is the fact that it has an organic final electron acceptor. So if we stop at the level of glycolysis with pyruvate, and that bacteria takes pyruvate and eventually converts it into an acid, an alcohol, or a gas, we know that fermentation is occurring. So we can kind of test for this fairly easy simply by looking for the presence of an acid, an alcohol, or a gas. And remember that fermentation just in general is a very low energy yielding process. So what we're doing this week is we're going to focus on the other 2 parts of the puzzle, which is aerobic and anaerobic respiration. So if you recall the characteristic of these 2, if we respire in general, we have an inorganic electron acceptor at the end, and in anaerobic respiration, it can vary. That final electron acceptor can be a variety of molecules. One example is nitrate. This is the example that we're going to look at this week. The example is nitrate can be reduced to other nitrogen-containing end products. And as you can imagine, the energy yield of anaerobic respiration varies depending on what that final electron acceptor is and the pathway that the bacteria use to get there. So finally, we have aerobic respiration. Of course, this is similar to what we do. Our final electron acceptor in bacteria that are aerobic respirers, their final electron acceptor is oxygen. And we take oxygen and we eventually turn it into water. We reduce it. We reduce it to water, and this, of the 3 options here, is the highest energy yielding process. So in terms of energy production, aerobic respiration is the standard. So if you kind of want to relate, I've talked, at least when we were in person, I talked a lot about thinking of energy as falling down a staircase, right? So at the top of the staircase is glucose. It's a very high energy molecule. And as glucose kind of gets broken apart, and it kind of bounces down the staircase, it's going to lose energy. We're stripping away energy from this glucose. And in bacteria that can only ferment, they can only take it a very small step of the way down this pathway. It can take glucose and turn it into pyruvate and glycolysis, and then that pyruvate gets processed into an acid, an alcohol, or a gas. And remember that this is only substrate level phosphorylation, very low energy process. However, if a bacteria is capable of respiring, that means that it can now take those electron carriers, the NADH and the FADH2, and take them over to the electron transport chain. And here they can do oxidative phosphorylation. That's the big energy payoff where we can get that good production of ATP going. So when we talk about these respiration processes, whether it's anaerobic or aerobic, it can really vary. All of the anaerobic processes have a range of energy. It just depends on what particular anaerobic anaerobic pathway we're talking about. But remember that the gold standard, of course, is our aerobic respiration ending with water. So here's the biggest drop in energy, glucose to water. This is aerobic respiration. That's where we're going to get the most amount of energy out of our metabolism. Or I should say more specifically, our catabolism, right? Our breakdown of glucose, our oxidation of glucose. Okay, so let's think about aerobic respiration. First, how can we detect whether a bacteria can aerobically respire or not? Well, one very simple way would be just simply to grow it. Under normal conditions, it's in contact with oxygen, it has to grow in the presence of oxygen, and therefore, if it can survive, then we know it's at least aerotolerant. It's able to survive in the presence of oxygen. We could also get a little bit more specific and look for the presence, grow the bacteria in the presence, or I should say in fluid thioglycolate broth, and look for growth at the top of the tube. Remember, that's where the oxygen is present. But both of these are very qualitative ways, and it doesn't give us a lot of good information about the bacteria. We could, like fermentation, if you think about what we did in fermentation, is we looked for end products. We looked for an acid, an alcohol, or a gas. So we could look for the presence of water, but that's going to be a fail because if you think you're adding water to media, that's already mostly water. So we can't detect these teeny tiny amounts of water production at the end of aerobic respiration in media that already contains a ton of water. So that's not going to work in this particular instance. But there is a couple things we can do. We can look for the presence of very specific enzymes that we know are part of the electron transport chain. This is what we're going to do in the cytochrome C oxidase test. And we can also look for that bacteria's ability to handle the toxic byproducts that go along with processing and using oxygen. If you recall way back, I think in Unit 1, we talked about this. When oxygen started to accumulate in the atmosphere in early Earth, like 2 billion years ago, that was very toxic to many of the organisms at that time. And we had this huge die-off of microorganisms at that time. And I really hope you guys can't hear that. My neighbor is still working on his car. I'm going to pause it and close my windows really quick. Hold on. Okay, so let's kind of keep continuing on here. Hopefully that's a little bit more quiet. And let's talk about the bacteria that did survive that accumulation of oxygen in the atmosphere developed very specific enzymes in order to handle that toxicity of oxygen. And the two enzymes are superoxide dismutase and catalase. So we can test for the presence of these enzymes, and that will give us a good indication that the bacteria that contain those enzymes are capable of growing in an aerobic environment. All right. Okay, sorry, I think I accidentally hit pause there. Okay, so let's take a look at our first test. And our first test is Lab 5.4, and it's a catalase test. Very, very straightforward test. Simply testing for the presence of the enzyme catalase. And this enzyme gives us a really good indication that the bacteria that contain this enzyme can tolerate the presence of oxygen. And so if you think about the electron transport chain in aerobically respiring bacteria, the electron transport chain is not perfect. It doesn't it's not like a linear pathway, and there are a lot of byproducts from this electron transport chain, and those byproducts are usually superoxide radicals, and those superoxide radicals are really damaging. They're like tiny little hand grenades that will damage the internal machinery of the cell if they're not taken care of right away. And the enzymes that do that are, of course, superoxide dismutase and catalase. So superoxide dismutase is going to take these superoxide radicals and convert them into hydrogen peroxide. And then catalase will take hydrogen peroxide and turn it into water and oxygen gas. So the presence of these two enzymes are basically protecting the cell from these really, really harmful chemicals that will irreparably damage the cell if left alone. So these, the catalase test is also a really handy test in terms of differentiating between two really important categories of bacteria. And that, of course, is strep and staph. So Streptococcus species are catalase negative. Staphylococcus species are catalase positive. So the test interpretation, easy on the one hand, because all you do is you add hydrogen peroxide and look for bubbles. It's really, really straightforward. But it can also be a little trickier than you might realize because, in fact, this is one of the tests, like if you were to do your unknown, if we were in lab and you were doing your unknown, this is probably the test that students screw up the most. Because they don't use a fresh culture and they don't use a positive and a negative control to see what bubble, like how much bubbles could generate. So sometimes like the wooden stick itself might produce a couple bubbles and they read that as a positive result. Or it might not bubble as vigorously as you expect it to and you interpret it as a negative result. So I would say that this is actually one of the tests where you really want to do a positive and a negative control to make sure that you're looking at those bubbles accurately. The other issue is that because you have to use a wooden stick because the hydrogen peroxide can actually react with the metal in your inoculating loop and create bubbles on its own. And so that would be interpreted as a false positive. So this is actually one of the tests, one of the few tests that you could actually do yourself if you wanted to. If you have a wound, you could put hydrogen peroxide on the wound and you will see bubbles. So that's the catalase. It's probably actually from your own body. We produce catalase as an enzyme. So you would see catalase activity in the formation of bubbles there. You could also, if you wanted to, I'm not necessarily recommending this, you could put a small amount on your gum line. So what I recommend you do is take a Q-tip and dunk it in hydrogen peroxide and then put it on your gum line. You're going to have lots and lots of bacteria there and you'll see the bubbles develop. Now this is super gross. It'll stink. And so I don't necessarily recommend it, but if you want to try it, you can. And then also remember that hydrogen peroxide is toxic, so you don't want to swallow it. So if you do this, just swish your mouth out with water and then spit it out in the sink after. I think there's actually, like some dentists, there are formulations of hydrogen peroxide that are kind of a lower concentration that you can use as a mouthwash. And then of course, they recommend that you spit it out and you don't swallow it like you would imagine. So, so there is some, hydrogen peroxide is used as a mouth detergent or disinfectant, I should say, in some cases. And you can also just try other things in your kitchen or your home. For example, yogurt has a ton of bacteria in it, so you could add, or any fermentation product, you have sauerkraut or kimchi or kombucha or miso or some type of fermented product. You could try to add some hydrogen peroxide to a small amount of that food to see if the bacteria that are in there are catalase positive. Unfortunately, yogurt will probably be catalase negative because most of the bacteria that are in yogurt are lactococcus or lactococcus species, and they're catalase negative. So it might work. You could also get more inventive and try your shower drain, right? Like the rim around the drain of your shower will for sure have bacteria there. You could add a drop of hydrogen peroxide and see if there's bubbles. You can try various parts. You can get some dirt from outside and so on, just to see where, if you can. And if you see anything interesting, let me know. I'd be interested to see if anyone came up with a cool catalase test. So if we were doing this in class, we would test two different bacteria, Staphylococcus epidermidis and Lactococcus lactis, as the previous slide, I think it is. Oh no, the previous slide. Lactococcus species are catalase negative. Staphylococcus species are typically catalase positive. So we see that result confirmed here. And again, this is a really vigorous result. This is a very, very bubbly result. We don't typically see it like that strong. And again, if we were doing this in lab, we would have a positive, actually these would be our positive and negative controls to confirm that that our test is actually working. All right, let's move on to our next one, our oxidase tests. So this is something, remember that in cells that can respire, and let's just narrow it down to aerobically respire, there's this assumption that their electron transport chains are all the same. You go to A and then B and then C and you're done. And that's actually not true. There's a lot of variety in electron transport chain, the specific components and the linkages in those electron transport. Now we have a helicopter. It's so noisy in my neighborhood. Oh my God. Okay, so the biggest kind of misconception that students have is that all the electron transport chains are the same, and that's not true at all. There's actually a huge variety of electron transport chains that bacteria can utilize to undergo oxidative phosphorylation of those electron carriers. So there are some commonalities. So for example, there's always an initial flavoprotein that accepts those electrons from, NADH or FADH2. There's always going to be some type of proton pump in that chain to pump those hydrogens across the membrane to create the gradient. And then I guess one that I didn't list here, of course, is that at the end there has to be something that is going to use those electrons to reduce oxygen to water. Now, if you look at the electron transport chain, some bacteria have what's called complex 4, so it's one last step in their electron transport chain, and some don't. And so what this oxidase test is doing is it's specifically looking for bacteria that have this complex 4. And complex 4 is also known as the enzyme is called cytochrome C oxidase. So we're going to test for the presence of this specific enzyme, the cytochrome C oxidase, by adding a chromogenic reducing agent. And so that sounds very fancy, chromogenic reducing agent. And simply what it means is that when it gets oxidized, it's going to turn color. So think about how like iron goes from gray to red when it becomes oxidized. And that's really what this chemical is doing. So the chemical is called TPP. It stands for tetramethyl phenyldiamine or so. It's some big, long, fancy word that we don't really care about. Let's just call it TPP. And it changes color. It goes from a clear to this really pretty dark purple color when oxidized. And so if cytochrome C is present, it's going to turn this nice, pretty purple color. And you need to read it within 15 seconds. Otherwise, you can get some secondary reactions that give you a false positive. So let's look at some results here. So there are a variety of ways to look at results or do this test in a lab. You could add the TPP directly to the bacteria on your agar, and the presence, like you can see, it'll turn purple in a positive bacteria here, and you won't get anything in a negative bacteria. You could also soak a cotton swab with the TPP and then rub it onto the bacteria to get that positive result. We typically do this method in lab. We basically soak some filter paper with the chromogenic agent, and then we apply some bacteria. And so this is the result that I want you to interpret here. If we were doing this in lab, we would use E. coli and Neisseria ceca as our 2 bacteria. So go ahead and interpret these results and record them in your data sheet for this lab. So far, so good, right? Well, let's talk about our tricky one. Our tricky test that we're going to talk about today is the nitrate test. And the nitrate test is going to specifically look at bacteria that can anaerobically respire and use nitrate as a final electron acceptor. So one pathway, one pathway of anaerobic respiration is utilizing an enzyme called nitrate reductase. And this enzyme, this is right out of your book, I think it's on page 307. Nitrate reductase is an enzyme that's going to take nitrate, NO3, and reduce it to nitrite. And so bacteria that are capable of doing this are capable of anaerobically respiring using nitrate as their FEA. But it's not quite so simple. It's not just kind of this simple one-step process. Once this bacteria converts nitrate to nitrite, it's possible that it can further take nitrite and further reduce it into other end products. For example, it can turn it into ammonium and then use ammonium to make amino acids. It can take nitrite and turn it into nitric oxide or nitrous oxide, and then eventually generate some nitrogen gas. In other words, we're taking nitrate and turning it into lots of other nitrogen-containing end products. We can't do this, actually. Bacteria can only do this. So let's look at some potential outcomes. We could have nitrate unable to produce nitrite. Right? So this couldn't happen. This pathway is not functional. We could have nitrate getting reduced to nitrite. We could have nitrate being reduced to nitrite, and then nitrite being reduced to some other type of nitrogen-containing product. And then finally, we could have nitrate being reduced to nitrite, and then nitrite to ammonium, and then ammonium to amino acids. So we have all these potential outcomes of this pathway. And it's important to point out that even though this is called the nitrate test, what we're really doing in the actual test is testing we're testing for the presence of nitrate. We're testing for the presence of this reduction product here. And the way that we do that is nitrite in solution turns into a very weak acid called nitrous acid. And nitrous acid is going to react with sulfonylic acid and naphthalamine. I think we just call them, we call them reagent A and reagent B. And when we add these two, I think we add it's like several drops of reagent A and several drops of reagent B. And as soon as we do that, if nitrite is present, it will turn this really nice, pretty dark red color. So in this step of the reaction, red color is an indicator of a positive result for nitrate reduction. All right, so let's go ahead and, let's ask ourselves before we go to the next step, which of these bacteria, let's just say that these are, 4 different bacteria, which of these bacteria have the enzyme nitrate reductase? So hopefully you can see, of course, because nitrate is being reduced in all 3 of these instances, these 3 all have that enzyme, nitrate reductase, but this one does not. Oh no, I need to, hold on, I'm going to pause the video. I need to plug in my computer or I'll die. Okay, I won't die. My computer will die. Okay, I'm plugged in. We're good. So, and I think this is where I left off. I'm all flustered now. This particular bacteria does not have nitrate reductase, so no nitrate reduction can occur. So again, we have red color would be a positive result. But let's look at this in a little bit more detail because this is tricky. Which of the following, ask yourself, in which of the above, so we have 4 different situations here, which outcome is nitrite present? Remember, we're testing for nitrite. So which of these 4 situations has nitrite present? So I'll give you a hint. It's only one. There's only one situation where we see nitrite, and that is this one. Here's nitrite. Remember, nitrate is being reduced to nitrite, and we'll see that red color. We'll see that red color develop. Here, it can't happen. We're still stuck at nitrate. In these 2 situations, we had nitrite for a brief period of time, but then that nitrite was further processed into other end products. So if we were to stop here, right, if we were to stop at this point and only add reagent A and reagent B, we would get a true negative for this result. We would get a true positive for this result, but we would get a false negative for this result and this result. And that's not good enough. That would mean our test isn't sensitive enough to detect nitrate reduction in if we were to stop here. So of course we have a further step. So what's that further step? So before I talk about the next step here, let's talk a little bit about zinc. Zinc is a metal, of course, and as like most metals, it's a really great reducing agent because it has a lot of electrons that it wants to give away. Remember from chemistry that metals make really good reducing agents because they love to give up electrons. So if we add zinc, it will reduce nitrate, if it's present, to nitrite. And then that nitrite will react with reagents A and B because they're already in the broth. Remember, we added them in the first step. And then now we'll get a red color. But here's the tricky part. Now, on the second step, the red color is now an indicator of a negative result for nitrate reduction. So which outcome, so ask yourself, of these 4 situations up here, which outcome still has nitrate present? Which of these 4 tubes still has nitrate present? So again, it's only one of the tubes. It's this one. Which one? This one. Nitrite is the only tube. This first instance, we still have nitrite present. And when we add zinc, what's going to happen? I have my animation off. What's going to happen? Let's see here. There we go. When we add zinc, zinc is going to reduce that it's going to reduce the nitrate to nitrite. The nitrite is going to react with reagents A and B, and now we're going to get that red color develop. And now it's a negative result. So in this step, in this one, we don't have any nitrate in the broth. So adding zinc isn't going to do anything, and we're not going to get a red color develop. Notice that we don't even have to add zinc to this test because we already have our positive result here. So you don't even have to add zinc if we already have that red color. So now we have a true negative. We have our true positive already from the just simply adding reagent A and B. And now because no red color developed even after the addition of zinc, we now have a true positive result, which is our accurate result. All right. Just an aside, I want to point this out. I usually talk about this briefly in lab. Nitrate tests typically include a Durham tube. I don't really like including a Durham tube, but it's in the book, so I thought I would mention it. To me, a Durham tube gives you very kind of, I don't know how you'd say it, muddy results. So you'll see gas production a lot of times in nitrate broth, but you don't know the source of the gas because almost all the time, almost any time you do this, the bacteria that you're testing can ferment in some way. And if your bacteria is a fermenter, you don't really know the source of the gas. And so if you don't know the source of the gas, there's really no conclusion that you can make from it. But the idea here is that if you're working with a bacteria that you know for sure is not a fermenter and it produces gas, you can assume that that gas is nitrogen and therefore denitrification has occurred. In other words, the enzyme or the bacteria is nitrate reductase positive. I don't see any value in it though. It doesn't give you a result that you can count on like the red color, like the addition of reagents A and B and zinc give you very clear results. Here, you have to know what bacteria you're working with, and if you're working on an unknown bacteria and you're trying to characterize it, you probably don't know if it's a fermenter or not. So the presence of a gas bubble really doesn't tell you anything. But I thought I would just point it out because it is in the book and it shows it in all the pictures. So I don't know, I just ignore it. Okay, so let's talk about some results here. So if we we're in lab, we would look at these three bacteria, Alkaligenes faecalis, E. coli, Pseudomonas aeruginosa, and we would also use an uninoculated tube as a really nice control to make sure, of course, that there's no contamination and that our reagents A and B and zinc are working properly. So if we were to add reagent A and B to these four tubes, we would get a red result with E. coli, and the three other tubes would remain negative. And if we added zinc, we would get a red color develop in the Alkaligenes faecalis and in our uninoculated control, but our Pseudomonas aeruginosa would remain clear. So remember, we don't have to add the zinc to the E. coli because we already have our positive result from that. So go ahead and interpret these results, write them down in your data table for Lab 5-6, and use these results to answer the questions I think it's 1 through 4 in your data sheet. So that does it for this week. Under half hour, not too bad. See you guys next week.